Happy Friday and welcome back to FirstThought Focus. Here at FirstThought, we are a life sciences focused expert network with a mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts, while also covering a wide range of topics that are relevant and important to our industry. You can find a list of our upcoming events and replays of our past events on cell therapies, CAR T, and the human microbiome on our website, firstthought.io/focus. In June next month, we will be featuring the second webinar in our series on the human gut microbiome as it relates to CNS disorders, and are also planning a post-ASCO recap event. We do want to hear from you. So if you have a topic you'd like us to feature in an upcoming FirstThought Focus series, please reach out. We'd love to hear from you. And finally, just as a quick reminder before we get started, we will be taking questions from the audience at the end of today's discussion. You can ask or submit your questions via email to hello@firstthought.io. Again, that's hello@firstthought.io. And I just wanted to thank those who submitted questions in advance of today's discussion. So with that, I'd like to turn things over to our moderator and veteran science journalist, Neil Canavan.
Hi there. Welcome to FirstThought, everyone. My name is Neil Canavan. Today's webcast is going to focus on one of the most elusive of creatures, and that would be the cancer vaccine. Now, I came to know something about this topic. Morgen, am I on screen or are you on screen? I seem to be seeing you. Oh,
I see you, Neil.
Oh, good. There we are. Hello, and there's Lisa Butterfield. Okay, so let's try again. So I came to know something about this topic in writing my book called The Cure Within. It looks like that. This is published in 2017 by Coldstream Harbor Laboratory Press. And in 2 chapters of this book, it describes the perils of vaccine development, in particular something called GVAX, which we will touch on today. If you'd like a free signed copy of this book, just send your snail mail to Morgen, and I will go up to UPS tomorrow. Now, joining me today for this topic is one of the top researchers in the cancer vaccine space and the IO space in general, and this is none other than Dr. Lisa Butterfield. Now, she is Vice President of Research and Development, Parker Institute for Cancer Immunotherapy in San Francisco. Dr. Butterfield is also Adjunct Professor of Microbiology and Immunology at UCSF. She's currently chairing an FDA Cell and Gene Therapy Committee. Prior to all this, she served as President of SITC, which is the Society for Immunotherapy of Cancer, and was a longtime Professor of Medicine, Surgery, Immunology, and Clinical and Translational Science at the University of Pittsburgh. And during all this, in her spare time, she has published over 150 peer-reviewed journal articles, was the editor of a recent book called Cancer Immunotherapy, Principles and Practices, and she's also in a band. We'll talk about that later. Now, Lisa, I was speaking to someone the other day, and he deals with healthcare investments, and I was shocked to learn he had never heard of the Parker Institute, where in my little world, Parker is a huge fat deal. So before we go to the cancer vaccines, if you could just give a rundown on what exactly Parker is and does.
Absolutely, delighted, and good to see you, Neil. The Parker Institute for Cancer Immunotherapy is now 5 years old, and this was started by our benefactor and founder and president, Sean Parker, the entrepreneur. And he realized the power of cancer immunotherapy and started the Institute with an enormous investment. And it was created in part by Jeff Bluestone as the founding CEO and president. And it's a collaboration vehicle. It's a hub with a network of spokes at the top academic institutions across the country of UCSF, Stanford, UCLA, MD Anderson, Dana-Farber, Memorial Sloan Kettering, and then we include laboratories. And including Bob Schreiber, Nina Bardwaj, Phil Greenberg. So it's really a who's who of cancer immunotherapy. When I first heard the announcement 5 years ago at SITC, I thought, that's the model. It's right-sized. It's going to create small clinical trials testing combinations that need to be tested now with multiple partners and deep translational science. And so I followed them for a couple of years, and they have some new data at AACR on CD40 with checkpoint blockade and chemotherapy in pancreatic cancer that's really making a difference. And it's a movement that allows really focused collaboration and in a framework of win-win legal considerations that allow everyone to work together. So it's really an amazing dynamic environment. There are 60 or 70 people that work at Pisces Central, and the scientific retreats are some of the most wonderful scientific meetings I've had the pleasure of attending.
Oh, that's really great. I mean, yeah, for me, it's like, I don't know, like going to Woodstock. And so you guys, anyway, it's an amazing place. So let's get back to the subject at hand. We're going to start with the most basic of basics. And so regardless of the type of the vaccine, be it heat killed or peptide or DNA or whatever, let's just start with the simplest possible thing. I inject something under my skin, and then what happens?
And then you've perturbed the system. There's going to be some inflammation. And some of those immune cells are going to come and see what it is you've injected, and they're going to react to it. There's going to be a direct flow of antigen. If there's whole antigen in there, there's going to be a local uptake of that antigen by antigen presenting cells. And the whole either antigen and or antigen presenting cells are going to traffic to the draining lymph node, and they're going to process and present that antigen and start talking. And they're going to talk to T cells, they're going to talk to NK cells, they're going to talk to B cells, all in different regions of the lymph node, and you're going to start the immune response. Okay.
Now, I was talking to Patrick Hugh a while ago. He's the CEO of Moffitt now. He's very big into cancer immunology. And he once told me that vaccines for infectious disease is easily the greatest medical advance of all time, had saved untold millions of lives. Now, the first problem with using a vaccine relative to cancer is your polio vaccine does not treat polio. It prevents it. So when we're talking about cancer vaccines, to my knowledge, none of them are being used in a preventative way, except for HPV, which is not strictly a cancer vaccine. But there are two that are approved. One's been around forever, called BCG. Another one's called Provenge, which was made by a company called Dendreon. BCG has been around so long, it doesn't even have a brand name. Those are just the initials of the guys who came up with it. So let's get a little bit more into mechanism. First, what is BCG and why does that work?
Well, BCG relates really all the way back to Coley's toxins. And it's
like 100 years ago.
Yes, some of the earliest cancer vaccination. This is a type of a way to awaken the immune response with some pathogenic molecules. And so I think of it as a very potent adjuvant. The immune system, as the point you've made, is designed to recognize pathogen. It's not designed to recognize self, but it helps if there's danger. And so if we can create the right kind of inflammation, the right kind of initiating signals, then maybe we can skew a system that's starting to go down a cancer progression supporting the cancer path and change that to a directed inflammation, what we call a type 1 immune response that pushes the immune system back towards a cancer fighting, non-cancer promoting type of milieu. So that's the way I think about BCG approaches.
Okay, just briefly, when people are reading papers about this sort of thing, they come across the word PAMP, and a PAMP may not be defined within the paper. Could you just briefly discuss what is a PAMP?
A pathogen-associated molecular pattern.
Good.
And the PAMP it's that signal. It's that there's a foreign invader, the immune response has to kick in, and it has to kick in now. So it's a pathogen-related signal, and there are some core differences between bacteria, viruses, and you and me. And the immune system can cue on those very, very quickly and very powerfully. So having a PAMP as part of your vaccine strategy, I think is a potent way to promote immune activation.
Okay, I did refer to just a few minutes ago HPV. It is associated with several types of cancer, cervical cancer, head and neck cancer being the most obvious. And there are therapeutic vaccines development looking at these settings. And that takes advantage of this idea of the PAMP. But does that work inform in any way when you're considering prostate cancer or lung cancer? Or that's just an entirely different beast?
Those are different beasts. The link, there's less linkage to pathogens being drivers. The thing about HPV is there's two proteins it carries called E6 and E7, and they are able to inactivate tumor suppressor genes that are key to cells killing themselves if they start to proliferate uncontrollably. So if you inactivate that, you jumpstart cancer by allowing cells that have been infected to just keep proliferating. So there are some keys about the tumor types that are specifically associated with the HPV infections that make those, I think, unique, and they're naturally more immunogenic, and they have a very different biology from, say, a head and neck cancer that came from smoking exposure.
Okay. So we're talking about antigens, and I want to jump into that. We want to choose the right antigen or antigens. And for this, I want to put up a graphic. Morgen, okay, there we go. We see here in the middle, we have oncoviral antigens, which apparently are ideal and can be used in multiple patients. All right, very good, yes. So for this, where would you place your bet here? If you want to go after, let's say, lung cancer, where in this chart where you be?
Well, for lung cancer, I would love to use a shared neoantigen. We're still evaluating cancer vaccine platforms. So when and individualized neoantigens are still a little hard to identify. There's a huge pipeline of all the mutations, mutations that are processed and presented, mutations that are processed and presented by that patient's HLA that are immunogenic. So, and it takes weeks and weeks to do that. So what would be ideal is if we could figure out mutated RAS, mutated MYC. These are mutated P53 common mutations that are new to the tumor, which makes them a little more like an infection. It's not something the tumor's been seeing and processing and presenting. It's unique, and it's not expressed anywhere else on any other cell. So if we could find a great platform to deliver immunogenic shared mutated antigens across tumors, that would be a big leap forward.
So yeah, I hear about neoantigens all the time. Everybody has all sorts of platforms. So when you look at a neoantigen platform, since predictive algorithms are proprietary and it's math, and you don't get to look at it, when you see these things, you shrug and say, oh, okay, whatever. And I guess I need to see some data. So with that idea that you can't really analyze how they derive this antigen, would private antigen data be interpreted any different from the shared antigen data if we're talking about neoantigens?
So two things. Your question first. There, I think it would be different because there would be, it's got such potential. It's not the potential of individual antigens in a patient that are going to be different from everyone else's individual antigens in their tumor. And so there's a lot of variability in how they can be harnessed and what they can do. And we're looking carefully for those tumor regressions in patients who have tumors, as opposed to extending the life of patients who don't have tumor yet, don't have tumor anymore are likely to recur. Oh, and we also gave PD-1. So a lot of the neoantigens have been tested in a very specialized way in order to enhance maximal benefit, but it makes unclear the role of the neoantigen. A shared neoantigen, I think, would be seen as a bit more promising with a bit more potential. But the point you've made about the algorithms to identify the neoantigens, this is one of the reasons I joined the Parker Institute, is a program we call TESLA.
Okay.
Because it's cool. And it's an acronym that you can read the paper. But the paper came out in Cell last year, and it's a consortium with many, many players, academic players and pharma partners who did the test. And they got the same sequence. They all ran their algorithms. The peptides were made from all the different algorithms and then sent to the academic labs to run the same assays and to test them for immunogenicity and processing and presentation. And how well did each of those anonymized algorithms do? And that came up with 5 aspects of these algorithms that across them are important and are in common between these algorithms, and that should be incorporated, I think, in all of them.
Okay, cool. Was Tom Schumacher part of that, I think?
I believe so.
Okay, yeah, all right. So let's say we've chosen an antigen. So the next step is we have to pick a modality to use to deliver this thing to the immune system. But before we talk about methods that are in play right now, I want to do a bit more of history, and I want to touch back on Provenge. I talked to the CEO of Dendreon, the maker of Provenge, just prior to the launch, and he showed his data. And I told him the science, I thought the science was really cool, but in my understanding of the mechanisms, I was puzzled as to why it didn't work so great. And his response to that was, yeah, me too. So this is a similar question to BCG, but kind of opposite. So what is Provenge? And why do you think the efficacy is so modest?
So Provenge takes an activated autologous blood cell and loads it with antigen and gives it back in about 3 days. And it's a mixture of cells. They don't do a lot of purification. Some people think of it as a dendritic cell, a potent antigen presenting cell. I don't, because they didn't make it the way most dendritic cells are made. It's got some of those components. It's also got a lot of lymphocytes. And so it's sort of it's part antigen presenting cell, and there's some activated lymphocytes that may or may not be playing a role. And it's a shared overexpressed antigen. So on your chart, it's one of the less favorable antigens. It's not mutated, it's not unique. And so the Kaplan-Meier plots of survival looked more like chemotherapy. It looked, like, the patients do a little better, but, you don't get, that curve, the famous immunotherapy curve with the tail.
Yeah.
Where, people were cured, but then there's the patients who seem to be cured and have really long-term demonstrable survival. So there's less of that. And so there's now a lot of effort thinking, how do we make this better, instead of having it be really seen as a standalone, and others have never incorporated it because the effect is not that powerful. So, and they've looked at mechanism and they've looked at biomarkers, and they haven't seen much to hang their hat on and build on mechanistically.
Okay, I'm glad you make that distinction about what exactly it is, because I talked to one of their scientists years later, and he's to this day still annoyed that people go, well, it's a DC vaccine. He goes, no, it's not. So we're going to touch on that method in a few minutes. But I want to go again into history. I want to talk about something called GVAX. This was developed in '93 by Glenn Dranoff, who's now at Novartis, I think. And the idea here was twofold. So for the antigens, you get the tumor cell from the patient. And for the immune system boosting part, you genetically engineer those cells with a cytokine called GM-CSF. Then you give the whole thing back to the patient. The patient's own immune system chooses the antigen to go after. Now, this drug in development since '93 has yet to be approved. People are still trying to do it. Is this the same underlying problem with Provenge, or is this more of a tumor microenvironment issue? How come this isn't approved?
Oh,
Or do we even know?
it comes and goes. It keeps coming back. And, you know, so it's tested, it looks good in early phase, it looks great in mice, you know, and then it fails, and then companies dissolve and come back. And then it starts showing up again, and it looks promising, and maybe in this context, you know, and then it doesn't, you know, quite pan off. So, but GM-CSF keeps showing up in screens of what's the best cytokine to add. And so GM-CSF has a role in promoting antigen presenting cells and supporting their growth and their differentiation to more mature cells, including dendritic cells. And so trying to figure out a way to incorporate GVAX, and I've, you know, it's, you know, combining it with listeria, expressing mesothelin in pancreatic cancer. That phase 2 looked good. So it's not, you know, the most beautiful dendritic cell. It's not a mature dendritic cell. It's not, you know, it's sort of its own thing. And I think it's weak. GM-CSF is an important signal, but when you've got a weak signal and you don't have groups of patients who responded completely and groups who didn't, so you can start looking at mechanism, you're left with sometimes we see something and sometimes we don't, and we don't know why, which I think really speaks to the entire field of cancer vaccination.
Okay, so you mentioned the pancreatic, and it just anticipated my next question. So Liz Jaffe at Hopkins, she was working on GVAX for quite a while. And I spoke to her about it, and she goes, yeah, you know, it's the weirdest thing that several of my patients from years ago are still alive. And, you know, pancreatic cancer, this is pretty dire prognosis for that. And in conversations with George Kukos at the Ludwig Center, he said a very similar thing about a similar approach in ovarian cancer. He goes, yeah, I've got patients to this day, but there's just no consistency to it. So where are we in predicting if someone will respond to a vaccine? I mean, just to enrich the enrollment or something.
The best data I'm aware of is about pre-existing myeloid-derived suppressor cell frequencies. And that's in both a preventative vaccine setting and in data from vaccine trials, where if you measured myeloid-derived suppressor cells and the patients had, you know, higher than whatever frequency at baseline, those were patients who were less likely to be able to respond to a cancer vaccine. So that's, you know, that's, I think, one of the best biomarkers, but myeloid-derived suppressor cell measures are largely phenotypic and not functional, so they're not the best yet. And they're not so robust that you'd actually deny enrollment to a patient based on it.
Okay, all right. So we've touched on dendritic cells and DC vaccines, and I want to put up another graphic. You published on this recently. So very simply, how does this work? And let's start with, how come you just can't shoot it under your skin like a flu shot? Why is this a preferred method of delivery?
I think all of these ex vivo strategies where you take the cells out of the patient, you soup them up, and then you give them back, work on the premise that the patient is immune suppressed, especially the cancer patient who is graduated to being a clinical trial patient and no longer getting whatever the standard of care is, that things have so advanced, their tumor microenvironment is set up, that our best hope is to get the cells out, do what we can, and then give them back. So this is adoptive cell therapy as well. So here, you take the blood cells and they look pretty normal on the surface anyway. And if you can culture them in GM-CSF as a growth factor and IL-4 to skew differentiation, and load them with purified forms of antigen, and this is, you know, sort of getting away from a tumor lysate where you'd be loading them with tumor antigens that you know about and don't know about, which is good, and IL-10 and TGF-beta and all these negative regulators. So you can have purified systems ex vivo and give the best cell back to the patient that you can give them. So that's why all this rigmarole, and all this rigmarole is actually, you know, $7,000, $8,000, you know, per patient for a course of these cells, as opposed to checkpoint blockade or adoptive cell therapy. So it looks complicated, but it's not, and it's not expensive either.
So let's go to more rigmarole, as you say. We have another graphic. And let me see, where am I? Oh, yes. So I lifted this from a paper by Patrick Ott, who's at Dana-Farber. So we'll note at the top line, there's the platform choice. And then there are the ways to load things on there. What would be your preference for how to load up your antigen?
Oh, would that I knew. So I've always favored dendritic cells because I think the dendritic cell knows a whole lot more than I do about antigen presentation. Okay. And that the dendritic cell, that's its role in our body. And if we just give it instructions, like pathogen instructions, like PAMPs or other signals, that we can tell it which way to go with this antigen we've just given it. And we've been following different markers on dendritic cells for a long time, and they don't really correlate with anything other than, yeah, that's a dendritic cell, but how good that cell is at what it does is something we're still learning about. I would not have said RNA about a year and a half ago, but the work of people like Ugur Sahin have worked and worked and worked at RNA in terms of packaging it and modifying it so that that's now turns out to be a pretty great way to give it with, 10 plus years of really focused development. I've given DNA, and DNA in primates, unlike mice, is pretty inert immunologically, so you have to really soup that up to get that to be stimulatory. Viral vectors, I've been a fan of some not so much because they actually replicate in or destroy or are pathogenic to cells like dendritic cells, but others are not. And so that's an easy way to include a pathogen signal in your, with your tumor antigen to try to push the immune system in the direction we want it. So there's, there are pluses and minuses to all of those. Just straight peptide vaccines without a dendritic cell or something else, I think have been less successful. They're great at getting cells in the peripheral blood and have been weaker at getting cells into the tumor where they need to be. So pluses and minuses, but those are my my biases and my read of the state of the field.
Okay, just for the benefit of our listeners who may not know, the two vaccines that are out there, Moderna and the one before Ugur Sahin from BioNTech and then Pfizer, these companies are cancer vaccine companies. That's how they get started, and they've been plugging along for years trying to do that, and this is their first hit, so infectious disease. So another thing I want to touch on, though, I see a lot of platforms that have something called the prime boost, and all vaccine platforms have multiple shots, but flu is one and done, and COVID hopefully is two and done. Why do you have to keep boosting?
We don't know. How often do you have to boost? What's the best boost interval? How many boosts? When can you stop boosting? Those are all great questions. And the studies that vary that number, the patients who do the best are there to get the most boosts. Did the boosts help them? I don't know, because some patients got 2 and some patients got 10. So we're pretty, and if you measure immune response after every boost, so all of the studies I've been directly involved in are 3, all the dendritic cell ones. And if you measure after each one, the kinetics of patient responses, some patients don't respond until that 3rd one, at least in the blood. Others after 2, others after 1. So I think our thinking is we want to make sure they're going to respond. So let's 2's minimum if it's really powerful and immunogenic. But let's go with 3 or so. It's all our best immunologic guesswork, I think.
It sounds more like art than science to me.
Yeah.
So Morgan, can I have that graphic back up for a second?
It's a data-driven guess.
Yeah. Okay. Okay. I want to note now the bottom part of this, pre-vaccination, and then the post-vaccination stuff we can do. Pre-vaccination, are you a fan of this? Do you think it makes sense? FLT3, I know it's used a lot. GM-CSF we've touched on.
Yeah, So there it depends on the platform. So if you're going to give something that does not involve an antigen presenting cell or something really immunogenic like a viral vector that replicates, maybe FLAT3 ligand is a good idea because you can systemically, if you deliver it systemically, you demonstrably systemically improve the number of precursors to antigen presenting cells like dendritic cells, or you can give it locally right where you're going to give your antigen. And that's probably a good idea unless it's already in your antigen.
Okay. We're going to touch on the right side in a few minutes because you have published recently on that. I was going to ask a generic question about, describe the roadblocks, tumor microenvironment, MDCs, whatever. But instead of asking a simple and maybe not very bright question, I reached out to a very bright person that I know in the field, who I will not name, but he was in Jim Gully's lab for many years at the NIH. And then he went to a cancer vaccine company for many years. I will not name the company. And altogether, he worked on the cancer vaccines for 14 years. And about 2 years ago, he quit. And now does adoptive cell therapy. He just didn't think he was going to ever crack that nut. So I asked him, okay, I'm going to be talking to Lisa. Do you have a question? He says, oh, do I ever? And so what he did was not really a question, but he sent me a bullet list. And he goes, this is what we have to do. And it was like, okay, antigen selection, mode of delivery, T cells physically getting to the tumor after they've been loaded, suppressive tumor microenvironment, T cell exhaustion, dose response, MHC loss. I skipped a couple, but the question was, how do we solve the problem of all these multiple variables in a clinical trial?
So one of the things I do pretty regularly every time I think about a new clinical trial is I either pull up the slide or I do my heads-up display of the cancer immunity cycle of Chen and Melman.
Yes,
which I love. So we need to check off all those boxes as you go around that cycle from presenting antigen to stimulating T cells to having them traffic to having them be able to kill the tumor. And so those are all important roadblocks. And I have noticed that people in this field do start getting pretty frustrated and like changed resistance mechanisms and tumor microenvironment mechanisms because that's where they need to put their energy. Nice. So I'm still in where does this fit in and how can we make it better place for some of that work. And so if we came in earlier, that might help, but that's going to be a while before we can come in earlier. So we need a reliable platform. And I think that's one of the things that GVAX and Dendrion's vaccine and some of these that are at least tried and true, so you know their strengths and weaknesses, and so you can maybe fit them in. And I feel that way about dendritic cells because that's a reliable immunogenic platform. We know it's going to prime immune responses in the blood, and it has the possibility of getting them to the tumor. But I think as we go around that cycle, the thing that we can't control, we can control priming, we can choose the antigen, we can get those T cells going. And if we can't get them going with a vaccine, we'll make a bag of cells ex vivo. But the tumor microenvironment, that is a very tough nut, and we come in late in the day. So we have to do something there to make these vaccines work better. And so we're, a lot of things in the pipeline that'll attack myeloid-derived suppressor cells. Some look pretty cool. And then think about just a hammer to the tumor microenvironment, like an oncolytic virus. Let's break open the tumor. Let's bring in an infiltrate. Let's bring in PAMPs, and let's really activate that tumor microenvironment and open it up. And then as we come in with our defined antigen, I like defined antigens because I want to know what I did, so I know the effect of what I did, as opposed to undefined antigens where the patient do that on their own. I don't know. So defined antigens disrupt the tumor microenvironment, but the key mechanism is epitope spreading. And unless we get epitope spreading to happen, we're going to have resistance. And so if you give a bag of T cells to one epitope restricted by one HLA molecule, you're going to be lucky if you get rid of everything. You have to come in with something else, or you have to induce spreading. And so we've seen that for a long time in cancer vaccines. The adoptive cell therapy folks see that now as a mechanism. So unless you can have an autoimmune response, unless you can initiate a response against the tumor that then leads to more antigens being processed and presented, and another round, in more rounds of T cells of different specificity, I don't think we're going to win. And those antigens, maybe we go in with the less desirable antigens that are shared across patients, but then if we can induce the spreading, some of those are going to be those patient-specific neoantigens, and maybe those are the ones that are really doing the job.
Oh, okay. You very nicely anticipated my next question, which has to do with oncolytics. Morgan, if I could have graphic number 4. This is a highly technical scheme. This was done for me by Rob Coffin, and Rob Coffin invented something called PVEC, which is an oncolytic virus treatment, which I think is herpes loaded with GM-CSF. And this is now approved, and I think Amgen had it. Doesn't work that great, but that's not the point of this. What it does illustrate rather crudely, though, is what's called an abscopal effect. So we shoot the vaccine in your leg, directly into a lesion, and then it through epitope spreading, one assumes, it goes all over there and gets your micromets wherever they happen to be. So there's a buzzword connected to this, and I think this aptly applies to this, which is in situ vaccination. There's a lot of platforms that do this. So sort of contrary to what you just said, I like this idea because you can't lose the antigen because you don't know what antigens are going after when you use an oncolytic. And I know you published on this recently with the vaccinia virus. So what was your experience with oncolytics?
So I think there's a lot of promise here. And what you've said, that abscopal effect in the spreading, and that is absolutely the key, because you can inject a tumor with anything and kill that injected tumor. Acetyl alcohol, ethanol, you can inject anything and make that tumor go away. If you don't have a systemic effect, you're nowhere. You could have injected anything. So and I think part of it is the ability for those viruses to replicate when they get in there. And that's been a little tough, and it's tough to show. And you can show it in, TVEC went in melanoma, and you can always get someone's melanoma metastasis and look at that. But in terms of, some of the bigger killers and the hard to biopsy tumors, if you could go in with a core needle, what's the chance you're going to hit the bit where the virus is replicating and know whether it was replicating there or not? So it's really hard to test in other tumor types. You just start inferring it because, you injected it over here and you have an effect over here. So that spreading is critical, and getting a decent dose of virus in with decent pathogen signals and decent tumor replication and lysis. So I think there's a lot of work to do, but that's I think been a limitation. And also intravenous delivery. It would be great if you could do intravenous delivery, and then there's a chance with tumor-specific replication of some of these viruses. Now you've seeded many tumors and metastatic sites with the virus that can start the ball rolling in multiple sites.
Okay, now I want to go on to combinations. Now you've published on this fairly recently. You used the checkpoint inhibitors, the famous checkpoint inhibitors. I believe you used PD-1 in your study.
It was ad hoc, but yes.
Okay, tell me what you did and what you saw.
So the trial was a dendritic cell vaccine engineered with an adenovirus for some PAMP signals, expressing full-length antigens, so it was not HLA restricted. And then it was a combination trial with interferon alpha. This is melanoma. Interferon alpha is approved. It's a cytokine that has been shown in a number of studies to amplify tumor antigen specific cells and also induce spreading. So we thought, so it was 2 arms. Everyone gets the vaccine, some get observation, others get 1 month of high dose interferon. So the time we started this study, ipilimumab, CTLA-4 blockade, had been approved. And so some of the patients had previously gotten checkpoint blockade, mostly IPI, and some had not as standard of care. Then people enroll on the study, 35 patients, everyone gets vaccine, half get interferon, and the study's over. There's no continuous vaccination. And so then you see what happened. And in some patients, if they recurred or didn't respond to the vaccine and interferon, they then got standard of care, which would be checkpoint blockade. So we had patients who had checkpoint blockade first, got vaccine, and then we looked at what happened. Others got vaccine first, we see what happens, and then they may have gone on to get checkpoint blockade afterwards as standard of care. So it was not a rigorous controlled study. It's ad hoc observation for the patient population we had. And what we saw is if you got checkpoint blockade first, you had a higher baseline frequency of tumor antigen specific T cells, but it did not set you up for success in particular. You didn't do better clinically. You didn't respond better to the vaccine. You just had a little bit higher frequency of some of these shared antigen specific T cells. We had 2 patients who got vaccine first, then checkpoint blockade, for whom we could get blood. And they had a 40-fold and a 100-fold increase in the number of vaccine specific cells. So that suggests, and this came out right after Samir Khalif's paper in a mouse model, suggesting the same thing, that vaccine first, then checkpoint blockade is something that can then amplify those effects. Checkpoint first, then vaccine doesn't particularly, doesn't look like an appealing sequence based on the data we have.
Circling back to infectious disease vaccines, they work because it creates memory. Is there evidence of memory cell creation in cancer vaccines?
There is, and that was actually in the news recently from one of the high-profile neoantigen-specific vaccine trials that was conducted. And you can go back in a number of studies with shared antigens and find those cells circulating, and it's whether the patients still have tumor, whether they are no evidence of disease. So there's certainly memory from a vaccine-induced response. And what these new studies showed that for neoantigens, they can also see a pretty good frequency of circulating cells on the watch, looking for recurrence of those tumors that have that matched antigen. So we have to see, if there's evidence that those cells are infiltrating. The blood is easy to look at, and we for sure see memory cells from vaccines in the blood months and years later. So what they're capable of doing, we have to keep asking.
Okay, I have one more of a planned question, then we're going to go to the questions from the audience. So considering all we've discussed, let's say you ditch Sean Parker and you go to Wall Street, and you're going to evaluate the latest vaccine platforms. What are the things you want to see in terms of the approach, and what indicators of efficacy in preclinical models do you want to see? Maybe biomarkers.
I'm going to look at my Chen and Melman wheel of life, the cancer immunity cycle, and I'm going to look for a vaccine platform that addresses multiple mechanisms of antigen processing, presentation, and T cell activation. I'm going to wonder what the phenotype of those T cells are, and if they can be tracked to see if they can actually traffic to the tumor and get in the tumor. The first thing I'm going to look for is that it's more than better than PBS, because I see that a lot. I see PBS, us.
Yeah. Oh, wow.
So better than GVAX, better than Dendrion's, better than something that can work. Even if it's at 5% or has shown anecdotal, these anecdotal things, what are you better than? And do you get T cells to the tumor? And if there are any pre-existing MDSC, so some of these synthetic long peptides to the viral antigens look great if there aren't a lot of myeloid-derived suppressor cells on board in the patient. If there are, suddenly that needs a big combination approach to have the same efficacy. So what does the platform do when there's established tumor, not 3-day established tumor, and what does it do when there's myeloid-derived suppressor cells around?
That was a terrific answer. You're going to get a call from Goldman Sachs. Morgan, let's go to questions.
Awesome. Thanks, guys. Again, just as a quick reminder, if you have a question for Lisa or Neil, please submit it to hello@firstthought.io. First question that we have for you here, Lisa, is about COVID vaccine success in the last year. And someone wanted to know if the success with these COVID vaccines has helped in your area of research at all.
I think it has. I think it's brought vaccine science to the fore. It's shown that the investment in cancer vaccination and understanding those mechanisms paid off in a very big way by making that platform as proof of principle of that platform, enabling it to change course and just change the sequence of the antigen and prove itself. So that was a proof that the investment matters and that vaccines matter, and that we need to just find the setting for a cancer vaccine, as opposed to the bar of preventing an infectious disease.
Here we go. Can you hear me now?
Yep.
Great. Also wanted to know from one of our listeners if you could shed any light on timeline for cancer vaccine approvals.
I think we need some better Kaplan-Meier curves of survival of patients. We've got now the ability in reasonable time to make a neoantigen vaccine. We've got the ability to better harness some shared mutated antigens. And we're learning more about what matters and doesn't matter about a dendritic cell vaccine and about sequence. So I think we've got a couple years and a few trials, a few carefully done trials testing strong cancer vaccine platforms with rationally designed combinations to find the winning combination that will allow that to take off, or prevention data in high-risk populations like colorectal polyps, pancreatitis.
Right. Okay. Thank you. Another listener wanted to know, I know ASCO abstracts were released recently. Are there any in the cancer vaccine space that you are particularly interested in, or you think we should be paying attention to?
That's on my weekend to-do list. I confess I have not scanned my ASCO abstracts yet. Sorry.
That's okay. That's all right. Another question, what is your opinion about tumor vaccines using dying cells ex vivo treatment with non-replicating adenovirus and peptides? Is this a possible strategy?
And the pieces of that again are, and that's 3 pieces in one vaccine?
Yes, so this would be using dying cells after ex vivo treatment with non-replicating adenovirus and peptides.
So my concern always, and my bias about killed tumor, is all the baggage it brings with it. And it brings a lot of metabolic baggage and bad cytokines and suppressive molecules. And I don't want that in my cancer vaccine mix. So, some sort of viral PAMP is nice. And I know more antigens can be better in terms of, there's that study that Jacques Bonchereau and Carolina Paluca and Joe Fay did a long time ago with 4 peptides on DC. And the more peptides you responded to, the better you did. So multiple antigens is a good thing. And so it would be great to be able to incorporate easily neoantigens, shared antigens, overexpressed antigens, but coming up with a way to do that doesn't include all of those suppressive molecules would be more to on my favorite list.
I have a sort of follow-up to that, kind of, sort of. Has it ever been, I know with checkpoint inhibitors, you can get autoimmune diseases. Has this ever been observed with cancer vaccines?
So we looked at that in my last study, especially because we used interferon alpha as a combination. And so we've asked we ran autoimmunity clinical assays like anti-DNA antibodies and that kind of thing. And we didn't see any evidence that routine measures of autoimmunity might set the patients up for either a better response to the vaccine or toxicity from it. So we looked for that evidence, and by those mechanisms didn't see it. But I'm still pondering that possibility of that tie-in of autoimmunity.
Thank you. Back to you, Morgan.
Thanks, Neil. One of our listeners, Lisa, wants to know what your favorite preclinical cancer vaccine technology is right now.
Favorite preclinical. I'm not a big mouse person. So thinking in terms of just like technologies, I'm seeing some oncolytic viruses that, incorporate a lot of things to them that I like. And I've liked adenovirus for a long time because it's such a stable known quantity, and that it's got good, you can put a lot of stuff in it. So a virus that can replicate in the tumor microenvironment, that carries some good stuff with it, that will not just by direct lysis break open the tumor microenvironment, but carries things with it, like maybe release of chemokines to draw cells into the microenvironment, or from the study that the Parker Institute did with CD40 agonism. CD40 is such an important molecule, and we've been trying to figure out how to incorporate it. And so, that's another, signal, like CD40 and OX40 and these costimulatory signals in the context of broken open tumor and maybe some chemokine expression. Those are some things that I've seen in sort of bits and pieces here and there that I'm excited about.
Great, thanks for that. Another question that just arrived in wants to know, what are your thoughts on co-formulations of cancer vaccines with other therapies? For example, co-delivery as opposed to delivery of one therapy and then the other, as you described in your trial.
We looked in vitro in my study at presenting, sometimes you see PD-L1 on the T cells, and that may be an activation story, but it may be an exhaustion story. So we looked only in vitro at engineering the dendritic cells to present antigen and secrete PD-1 antibody at the same time in place. And we downregulated PD-1, and we didn't improve the activity of those cells. So, one in vitro experiment. But I think this cycle needs steps, and it's complicated by the fact that it's been going on for 10 years or so, before we get there. So I think we need to do things at least sequentially to an extent. We just don't know the sequence, and with humans, we're coming in different stages of their disease progression, and so it's hard for everyone to be level set to be able to test the sequencing. So I think sequencing is important. I don't think we can give all of those immune signals and all of those antigen signals at the same time. But it's going to be a tough nut to crack in humans.
I see we have 3 minutes left. We've gone through all the questions in the queue, but I do have one additional question. For listeners, Lisa has her fingers in many immunotherapy pies, including a lot of diagnostics and including imaging. And I was wondering, has there been any progress lately on imaging that helps you determine whether or not a patient will even respond?
So this is another reason I joined the Parker Institute, and that's things like MIBI and some of these amazing multiplex tissue imaging technologies. So there's data, this is a big data year at the Parker Institute. A lot of investments in early studies are all coming to fruition this year. So ask me next year. But in terms of technologies, this, being able to have these really integrated pictures, and instead of CD8s and FOXP3, because FOXP3 must be Tregs, unless it's not. So looking at these limited markers, I think maybe have led us down some paths, but having these incredibly multiplexed images of the real deal, of the tumor tissue before and after treatment, I think we're going to learn a lot in the next year from these.
Oh, great. All right, so we're going to wrap up, but first I want to make a big plug for something that's been mentioned several times in this call, and that is the Society for Immunotherapy of Cancer. We are going to meet in person with faces and everything this November in Washington, D.C. I encourage anyone who's interested in this topic to join. Lisa will be there. She will probably be speaking. We have one last graphic to show you that example of this. Lisa ran a panel on Immunoscore, and she was moderating. And you would think Immunoscore is not that interesting, but wow, I had such a good time at that session. So please come to DC, listen to Lisa talk. She might also sing. It's a good time had by all. And Lisa, thank you so much for joining us today.
Thank you so much, Neil. Thank you, Morgan. This has been a treat, and I look forward to seeing you at SITC.
Splendid.
Thanks, everyone. Have a good afternoon and weekend.